Hello children, we are discussing the chapter 2, that is biological classification. In this chapter, in my previous video we had studied the various classification system which were used earlier and the one which we are using right now. Now we will study the first kingdom, that is kingdom Monera, in detail. As we all are aware that the organisms in kingdom Monera are prokaryotes, are unicellular prokaryotes. What do you mean by prokaryotes? Pro means primitive and karyon means nucleus, means primitive nucleus. They have a primitive kind of cell. That means the cell does not contain any membrane bound cell organelles. They only possess ribosomes. The nuclear material is not enclosed within a membrane that is known as nuclear membrane. That material is directly present in cytoplasm and that is known as nucleoid. Prokaryotes does not have special kind of various enzymes enclosed in any certain membrane. The enzymes or every material which is present in the prokaryotes is directly present in cytoplasm. Usually the prokaryotes means bacteria. It has other organisms and bacteria, but mostly when we talk about the monerans, we are talking about the bacteria. Kingdom monerans or the monerans lives almost everywhere. So their habitat is usually they are found everywhere on the planet Earth. They are found in the most harsh conditions of the environment of the planet Earth. That is in the hot springs, Antarctic region, in the marshy meadows. Everywhere on the planet Earth they are found. They are found in the air, they are found in the soil, they are found in the water. So everywhere they are present. Their size is quite small. In fact, they are one of the most simplest and the smallest living organisms, as they are composed of prokaryotic cells. Next, monerans will study in 4 groups. The first one is archaebacteria or archae means old. They are the old form of bacteria and they are the ones which are found in the most harsh conditions of the earth. They have further differentiated into three divisions: methanogens, acidophils and the halophiles. That we will study in detail later on in this chapter. The second is eubacteria. Eubacteria are also generally regarded as bacteria. Whenever in our casual talking we say a term bacteria, that means most of the times we are talking about eubacteria. In our study as well, whenever we talk about the bacteria, that means we are talking about the eubacteria. Eu means advanced, the evolved bacteria. Third is, we are going to study the blue-green algae, that is cyanobacteria. One special thing about those bacteria is they are photoautotropic. They can synthesize their own food by the help of sunlight, as they have a blue-green color. That means they contain a colored pigment known as chlorophyll, which traps the sunlight, which traps the solar energy and then convert that solar energy into chemical energy as food. And the fourth class of this moneran kingdom is mycoplasma. It is the only group, it is the only class which does not have any cell wall. Usually all the monerans possess a cell wall and that cell wall is made up of peptidoglycans, but the mycoplasma, they are the smallest one as well among all these 4. These do not contain any cell wall. Next, we will study the bacteria or the monerans on the basis of their shapes. So the next topic of discussion is shapes of bacteria. Bacteria usually have 5 kinds of different shapes. The first one is spiralia. That is spiral shape. Certain bacteria possess that kind of spiral shape, and those type of bacteria are known as spiralia. The example of spiralia shaped bacteria is Treponema pallidum, which is the causative agent of sleeping sickness. The second is bacilli. Singular is bacillus. Bacillus means rod-shaped or oval-shaped bacteria. That is. These are the most common form of bacteria found in nature. So whenever usually we talk about the bacteria, the most common shape of bacteria is bacilli or bacillus. Third is the circular shape, that is coccus. Round shape. The next one is thread-like. That is less spiral form. That kind of thread-like bacterial shape is known as mycelium. So these are the various shapes on which the bacteria are found in nature. And the fifth is vibrio shape. Vibrio means comma. So the shape of bacteria is like this, comma shaped bacteria. An example of vibrio shaped bacteria is Vibrio cholerae, which causes cholera. So on these shapes, the bacteria are differentiated into various structure or various names or various genus. Next, watch it. This cocci shape, if there is only one single structure, that is known as monococcus. This same rule applies for the bacilli shape as well. If there is only single rod shaped bacteria, that is known as monobacillus. If there are two circular rings, then that is referred as diplococcus. That means two. And if there is a straight chain of circular rings, bacterial cells is present, then it is referred as streptococcus. And if those bacteria are found in the form of clusters, then that is known as staphylococcus. The same rule applies for the bacilli. If it is single rod, then it is known as monobacillus. If the two rods are attached to each other, that is known as diplobacillus or dibacillus. If the rods are arranged in a straight chain manner, then it is known as streptobacillus. And if the rods are found in the form of clusters, then it is referred as staphylococcus. Now we'll study the next structure is flagella, which is a prominent feature of monarins. The monarins or the bacteria have a flagella which is unistranded, single-stranded, and a very long structure. Some flagella are even more longer than the cell of the bacteria. On the basis of flagella, the organisms, or you can say the bacteria, are of two types. First, if flagella is present, then they are referred as trichus. And if flagella is absent, then they are referred as atrichus. So the bacteria which possess the flagella are known as a trichus, and the bacteria which do not have a flagella are referred as atrichus. Now we'll discuss the structure of flagella. Flagella is basically embedded like this in the membrane. This is cell membrane, and as we all are aware that the bacteria has a cell wall over that cell membrane. Flagella has three parts. First part is basal body, which is the base. The second part is hook, and the third part is filament. This basal body is composed of 4 rings. Which are attached with each other by the help of a rod. In gram-positive bacteria, these rings are only 2, and in gram-negative bacteria, the 4 rings are present. These rings are named as L, P, S, and M. In gram-negative, all the 4 are present, and in gram-positive, only S and M ring is present. These rings are attached with each other by rod. Out of these 4, the L and P are present in the cell wall, are embedded in the cell wall, and S and M are embedded in the cell membrane. So these are the 4 rings L, P, S and M, out of which the S and M are embedded in the cell membrane, the lower layer, and the upper ones, L and P, are embedded in the cell wall. This whole part is known as basal body. Then over that basal body they have a broad structure. You can see this structure, this one is broader than the rod. This is known as hook. So this part is known as hook. And from that hook a fibrous like structure is initiated, is floated, and that is known as the third part, that is filament. So these are the 3 basic structures of a flagella, that is basal body, hook, which is thicker in diameter than the rod, or the connection between the rings, that is known as rod, and the filament. So this structure, flagella, is made up of special molecules, special protein molecules named as flagellin. These flagellin molecules are attached with each other in the form of a cluster. And that cluster makes a flagella. So flagella is composed of a protein named as flagellin protein. On the basis of position of flagella, the bacteria are of different type. The first one is, if flagella is present at the single side, at the one tip, that is known as monotrichous. If the flagella is single and present on the both ends, then it is known as amphitrichous. So if flagella is present on the one end, either on this end or either on this end, on one end only, then it is referred as monotrichous. If flagella is single and present on the both ends, then it is known as amphitrichous. Amphi means both, 2. If flagella are many and present as a cluster on one side, either on this side or on that side, then it is known as cephalotrichous. If on the both ends the cluster of flagella are present like this, here only one side it is present, either on this or either on that, here on the both side, then it is known as lophotrichous. So if flagella is one and on either of the one side, then it is known as monotrichous. If flagella is on the both sides, then it is known as amphitrichous. If the cluster of flagella is present on the one end, then it is known as cephalotrichous. And if the cluster is present on the both ends, it is known as lophotrichous. And if the flagella is present all over the body, then it is known as part-2. So, if flagella is present all over the body, then it is referred as part-2. Now we will discuss the differentiation of bacteria on the basis of a staining that is named as Gram stain. Gram staining was discovered by Hans Christian Gram. That staining differentiates the bacteria into two forms, that is Gram positive bacteria and Gram negative bacteria. Which stained by the Gram stain are known as Gram positive, and which do not stained by Gram's stain are known as Gram negative bacteria. The basic difference between their composition is in their cell wall. Let us suppose this is the cell membrane. In case of Gram positive bacteria, there is a thick cell wall present over the membrane. This inner is the cell membrane and this one is cell wall. The cell wall of Gram positive bacteria is made up of peptidoglycans. But along with those peptidoglycans, there is an acid present in that membrane, and that acid is known as teichoic acid. This acid basically binds the layers of the peptidoglycans, and due to that teichoic acid, the thickness of this cell wall is quite high. Whereas, in case of Gram negative bacteria, obviously cell membrane it would be there, the cell wall is thin. Here the cell wall is thick, but here the cell wall is thin. Cell wall is made up of peptidoglycans, but teichoic acid is absent in case of Gram negative bacteria. Over that cell wall, there is another layer present in case of that Gram negative bacteria, and that layer is made up of, this outer layer is made up of phospholipids, polysaccharides, and lipoproteins. So in case of Gram negative bacteria, cell wall has another outer wall made up of polysaccharides, made up of lipoproteins and phospholipids. So these are the materials which make the outer layer over the cell wall. That is the basic difference between Gram positive and Gram negative. Rather than this, Gram positive bacteria has porins in it and they do not have porins. Its cell wall or its cell wall's thickness is 20 to 18 nanometer, and its thickness is approximately 8 to 12 nanometer only. The technique of staining of Gram negative bacteria starts with fixing the bacteria on a slide. Heat fixes the bacteria on a slide. By the help of heating, fix the bacteria on a slide. Then pour few drops of crystal violet dye over it. Place the dye for 30 to 60 seconds over it. Then rinse it with water so that excessive dye may be removed. After rinsing or washing it with water, pour few drops of iodine dye over it, or iodine solution over it. It is also known as Gram's iodine solution. Leave that solution also for 30 to 60 seconds or 30 seconds to 1 minute. After that, rinse it with acetone or alcohol. For this rinsing, you can take the 90% acetone solution or 90% alcohol solution. After that, you will observe the bacteria turns purple. After the iodine solution, the bacteria turns purple actually. The whole bacteria turns purple. But when you rinse that purple bacteria with acetone or alcohol, few of them will still remains the purple color with it and few will become colorless. अच्छा, तो जो यहाँ टोटल पर्पल कलर आया उसमें से कुछ कलरलेस हो जाएंगे और कुछ अभी भी क्या रहेंगे, कलर. The bacteria which remain purple colored are gram positive bacteria and which changes their color are known as gram negative bacteria. When we pour few drops of saffron in a red color dye over them, then they change their color to red. So these are the bacteria which shows negative gram staining and these are the bacteria which shows positive gram staining. The reason of that is, in case of positive gram bacteria or the gram positive bacteria, positive gram staining bacteria, this iodine solution is mixed into, is synchronized with the cell wall which is made up of peptidoglycans. But in case of gram negative bacteria, the outer layer over the cell wall which is made up of lipoproteins does not allow the iodine solution to goes into the cell wall. That is why when we rinse the outer layer that is made up of lipoproteins with the acetone and alcohol, the dye is removed away. And the saffron in the dye is the only dye which tends to color that outer membrane or peptidoglycan membrane into red color. Now we will discuss the respiration in molecules. Respiration is the oxidation of food to produce energy in a cell is known as respiration. Respiration is basically of two types: in the presence of oxygen or in the absence of oxygen. In the presence of oxygen is known as aerobic respiration and this one is known as anaerobic respiration. Aerobic respiration include following steps. The first one is glycolysis, which occurs in cytoplasm, in which the glucose, the carbohydrate substrate, is broken down into an acid known as pyruvate or pyruvic acid. The second step is Krebs cycle, which requires certain enzymes, and those enzymes are present in mitochondria, in the matrix of mitochondria, but the prokaryotic cell does not have any mitochondria because it does not have any membrane. Any specific membrane bound organelles are absent. So the enzymes which are required for the glycolysis and Krebs cycle are present in cytoplasm only. Third, ETC, that is electron transport chain, that we will study in detail in third unit. And the fourth one is oxidative phosphorylation, which ultimately forms ATP, the energy currency of the cell. So all these processes are required in aerobic respiration. Both these require certain kind of proteins, and those proteins are present in the cytoplasm in case of prokaryotic cell. In the cell membrane, there are specialized structure present which are known as mesosomes. Let us suppose this is the cell membrane over which there is a cell wall present. This cell membrane has certain inner folds in case of monoderms. In case of bacteria, those inner folds are known as mesosomes. These mesosomes are of two types. One is lateral and another one is septal mesosome. Those will perform separate functions. The lateral mesosomes are connected to the nucleoid and the plasma membrane, whereas septal are not connected to the nucleoid. So these mesosomes possess the proteins which are required for electron transport chain and oxidative phosphorylation in case of prokaryotic cell. Anaerobic respiration, glycolysis is same here. First, the carbohydrate substrate, that is glucose, is broken down into pyruvate or pyruvic acid in cytoplasm. Rest, after that, there are three processes for different substrate. The first one is for the carbohydrates, hydrates, which are broken down into alcohol, and that is known as fermentation. The second process is regarding the proteins, and that is known as putrefaction or putrefaction, in which the proteins produce energy in the absence of oxygen by the activity of certain microorganisms. The last one is decaying, in which the bacteria uses the anaerobic mode of respiration and converts the simple organic material into inorganic material. So these are the three various processes which are used in anaerobic respiration. On the basis of these two modes, that is aerobic respiration and anaerobic respiration, the monerons are further divided into two types. The first one is obligate aerobes, and the second one is facultative aerobes. What are obligate aerobes? The bacteria or the monerons which requires air to produce energy, and in anaerobic conditions they get killed. That means basically they are aerobes and can killed in absence of oxygen. They require oxygen for sure. Whereas facultative aerobes are actually anaerobes, but they can survive aerobic conditions as well. So these are actually aerobes and get killed in anaerobic conditions. The example is Bacillus subtilis. Facultative aerobes are actually anaerobes, but they can survive if oxygen would be present. In the absence of oxygen, they will survive obviously. But if the oxygen is present, then that presence of oxygen does not harm them. So these kind of aerobes are known as facultative aerobes. The example of facultative aerobes is halophiles. Likewise, anaerobes are also divided into obligate anaerobes and facultative anaerobes. Obligative anaerobes are actually anaerobes and get killed in the presence of oxygen. The example is Clostridium botulinum. That bacteria get killed in the presence of oxygen. They cannot tolerate the presence of oxygen. So the second one is facultative anaerobes. Actually they are aerobes, but they also can be survived in an aerobic condition. That means they use oxygen to produce energy, but if somehow or if any environment, due to any environmental change, the oxygen is not present, then also they can survive. So these are known as facultative anaerobes.